So as we were saying, calcium, magnesium, and zinc can also act as cofactors for enzymatic reactions. Remember, a cofactor is the non-proteic component of an enzyme. So, for example, zinc is involved in speeding up the reaction of conversion of CO2 into carbonic acid via carbonic anhydrase action. Sodium and potassium are involved in generation of the action potential. Calcium, sure, secretion and action of neurotransmitters and everything else. Muscle contraction. They also are buffers. I'm repeating these things not because I've got a working memory impairment. I repeat these things because they're important. So I do it on purpose, like to hammer always on the same concept that are important. So bear with me and get these things through, basically. Yeah. So yeah, there are certain ions such as proteins, the various phosphates like the hydrogen phosphates, monohydrogen phosphates, or bicarbonate, which work as pH buffers both intracellularly and extracellularly. They're also involved in primary and secondary active transport, by sodium potassium ATPase and glucose co-transport. So, sometimes glucose uses the gradient of sodium to move against its own gradient. So moving according to the gradient of sodium to move against the gradient of glucose. We will see how this is important in the gastrointestinal tract function. And of course, osmosis. Electrolytes and proteins mediate the movement of water through a semi-permeable membrane. We talked about this in the first lecture, and I will talk about it more in this one. So in terms of active primary transport, you perfectly know how the sodium potassium ATPase pumps are important in maintaining ionic gradients by pumping sodium outside of the cell and potassium inside of the cell and avoiding the leakage of sodium in and potassium out that would lead the cell to die. Sodium ATPase pump use up to 75% of the ATP of a cell in neurons, around 30% in a bacteria, and something between these two values in any other cell of the body. So you can tell how fundamental this protein is. Also, we do have secondary active transport, where the electrochemical gradient of one ion is used according to the gradient of that ion to aid the transport of another substance against their own gradient, such as glucose from the interstitial fluid inside the cells. So it's, for example, very important for glucose reabsorption in kidneys. Once again, the secondary active transport in the luminal or apical membrane in the intestine, thanks to the sodium glucose transporter 1, allows to move glucose that has a very, a lower concentration in the lumen than it has inside of the cell against its own gradient. By using the higher gradient of sodium from the lumen to inside of the cell. So basically, sodium entry drags other molecules such as glucose. This is not really exact. Let's say that the energy arising, the free energy arising from the movement of sodium from the lumen to the intracellular space of the intestinal epithelial cells provides the needed energy to move glucose against their gradient. So we talked about these already in our first lecture in the first term. We're going to talk about these again. The plasma membrane allows transport through different manners. So non-polar and small polar molecules can freely diffuse through the bilayer phospholipids. So polar molecules that are smaller than glucose that doesn't go through can go through the plasma membrane. So water can freely go through the plasma membrane. Fatty acids can move through the plasma membrane. Hydrophobic drugs can move through the plasma membrane. Steroid hormones can move through the plasma membrane through the bilayer of phospholipid. Larger molecules, or such as glucose, larger polar molecules such as glucose, or electrically charged species such as ions or electrically charged molecules to go through the plasma membrane according to their gradient through channel proteins. Molecules can move through the plasma membrane through carrier proteins, for example, facilitated diffusion of glucose, or against their own gradient, known as carrier proteins for active transport. And while the diffusion, whether it's passive or facilitated through a carrier or a channel, uses as a source of energy the electrochemical gradient or the chemical gradient of the species under investigation, under observation, the active transport transports that species against their chemical or electrochemical gradient. And the energy for this process is provided by the hydrolysis of ATP. Leads us to the process of diffusion in more general terms. So diffusion is the process by which molecules move from a compartment where there are more molecules to a compartment where there are less molecules, and the two compartments are separated by a semi-permeable membrane. Now, the movement of molecules in compartment A is Brownian. So we don't know exactly where these molecules are moving. We do know that they are moving. They're moving linearly, they're vibrating on themselves, they're rotating on themselves. So there are three types of movement. And these movements are stochastically distributed. They're random. But what we do know is that the higher the concentration of a species, the higher the probability that two species will collide with each other. And by colliding with each other in a random way, if we do have a higher number of collisions in A than we have in B, and the species cannot go on top, cannot go on the left, cannot go on the bottom, then there will be only one possible way they can go, which is from A to B. But also molecules present in B can theoretically move from B to A. But the total number of collisions that the particles have in the unit of time in A, they are more than in B. So there will be a driving force coming from the sporadic collision of molecules in A that is higher, stronger than the overall force that pushes particles going from B to A. So the net rate of diffusion is from A to B. You can kind of figure it out, right? If you're all crammed into a class, and they tell you now is the moment for you to leave, and you have to go through the bottleneck of the door, right? You will bump into each other, and you will tend to get out of the class and disperse yourself and occupy as much space as possible. That's how molecules work. So the driving force of diffusion is the concentration gradient. But if we're dealing with the electrically charged species, we do have two driving forces. One, chemical gradient, because it's still a chemical species, but also an electrical gradient. Let's stick to the concentration gradient and let's see how diffusion works. Stick defined a law of diffusion, where J is equal minus D times A times the very small concentration divided by the concentration in distance, where J is the rate of diffusion, so the net amount of moles that move from one compartment to another in a unit of time. D is known as the diffusion coefficient, and basically it depends on the intrinsic features of permeability of the semi-permeable membrane. You know, not all the semi-permeable membranes are plasma membranes. There are synthetic semi-permeable membranes that have different D values, and the minus sign indicates that the movement follows the concentration gradient. A is just the cross-sectional area, is a surface. Obviously, the larger will be the surface of the semi-permeable membrane, the higher will be the rate of diffusion. While the difference in concentration is what it is, the difference in concentration expressed in molarity or osmolarity or whatever it is. And the distance is the distance. So it is the distance between the two compartments.